This is Jim Schnabel. I am doing a series of talks from the ninth edition of Robbins, the book of pathology by Robbins and Cotran. And these are a series of talks which deal with general aspects of human pathology, not covering specific organ systems. That would be a more intense, directed field of study, but these are just broad general concepts, and we'll be spending the next few talks on this particular subject. The first is to look at some general definitions. The term pathology means the study of disease, and the human genome, based on the Human Project, has 3.2 billion base pairs. There are 20,000 protein coding genes, and about 80% of the human genome is involved in protein binding and regulation. There's additionally DNA variation, whereas a single nucleotide polymorphisms, which have variants in single nucleotides. There are also random variations involving coding regions in 1% of cases, and this is a weak effect on disease susceptibility. There are also copy number variations, which can involve thousands to millions of base pairs. 50% of these could occur in coding sequences, and these have an unknown effect on disease susceptibility. There are epigenetic factors. Proteins and nucleoproteins are involved in genetics. Among these are histone and histone modifying factors, which help to coil and uncoil DNA. The coiled is the inactive version, uncoiled is the active version of DNA. And this facilitates gene expression and allows transcription from DNA to RNA, and you can have these bases methylated, acetylated, or phosphorylated. There's also microRNAs. These are small RNA sequences which combine with proteins to induce messenger RNA cleavage or repress translation from RNA to protein. There are also long non-coding RNAs. These bind to DNA and they restrict RNA polymerase access to the gene. There are also elements of cellular housekeeping, and there are highly specialized functions to the cell. So if we look at this, the cytosol compartment is about 54% of the total volume, mitochondria 22%. You have endoplasmic reticulum, rough and smooth. The nucleus is 6%, and the other components, endosomes, lysosomes, and peroxisomes, are about 1% of the total volume. So this is a schematic of the cell with the rough endoplasmic reticulum and the centrioles, which are involved in contraction functions. The mitochondria are out here. There are peroxisomes, which are involved in degradation. The cytoskeleton causes a form of the cell to have a particular form. This is important in hemoglobin, for example, to increase surface area. You have the nucleus and nucleolus, which is the RNA center. It usually shows as red on hematoxylin eosin, and then external to the rough endoplasmic reticulum is smooth endoplasmic reticulum. There are membrane markers, phosphatidylinositol and phosphatidylserine, which are located on the inner membrane layer, bilayer. These are involved respectively in second signal propagation, and phosphatidylserine for apoptosis, which is programmed cell death. Glycolipids and sphingomyelin are on the extracellular face, and they perform structural and receptor function. So when we see this schematic, you have the external and the interior cytoplasm. Phosphatidylcholine is mainly on the outer aspect, phosphatidylethylamine on the inner, phosphatidylserine on the inner, sphingomyelin mainly on the outer. Lipid rafts, which are areas of lipid which literally can float through the process of the structure of the cell, with the inner component here, which are the hydrophobic areas, and the hydrophilic centers are on the external aspect, with usually a charge that causes a form of energy dynamism. Glycolipids are involved in cell markers and recognition, self versus non-self. Phosphatidylinositol is seen on both faces, and cholesterol can be on both faces. They're membrane carriers. These are gases which diffuse across the nonpolar membrane. Water is transported by aquaphorins. They're polar molecules. These are small molecules transported by channel proteins and specific carrier proteins, and these maintain electrical potential difference, which help drive the transport. For larger molecules, you have endocytosis, and this is driven by an organization of transmembrane proteins, and they assemble into a transit form. So this is a schematic showing these various mechanisms where you have a specific, the extracellular area and the cytosol matrix, the membrane and bilayer here. You can have an energy component with a carrier to pick up the entity in question. This is an active process. You can also have specific channels. These are like ion channels, for example. And the cytosis can be calvili mediated, where you have calvilin, which recognizes a substance and basically produces an outpouching and fusion of the membrane with these substances located. There are also receptors, which have receptor-mediated endocytosis, which bind to the area in question. And there's a coated pit, which also is extruded into the cytosol, and you get transformation to endosomes, early and late forms. And then these get reconstituted as lysosomes. For microbes, the phagocytosis is particularly important in infection-mediated immunity, and phagosomes and phagolysosomes produce an undigested residual material. This is extruded. Transcytosis can also occur. You get no alteration of the material in that formulation. The cytoskeleton, there's an actin, which is a component of muscle. Intermediate filaments are useful for establishing the cell of origin. They include desmin, glial fibrillary protein, vimentin, and cytokeratins. These are used as cell markers in pathology to determine origin of cells. Microtubules, you have facilitation of movement within a cell. There are also cell-to-cell interactions, tight junctions, which have a high resistance barrier to transmembrane movement and also maintenance of cellular polarity. There are also anchoring junctions, which are desmosomes, and these anchor cells to each other. These can be tight junctions, as you would see in the epidermis, with desmosomes connecting to squamous cells. There are communicating junctions, which mediate passage of chemical or electrical signals, and biosynthesis also occurs. You have protein synthesis in the rough endoplasmic reticulum, and these move through the smooth endoplasmic reticulum into the Golgi apparatus for protein modification. There's also degradation, which you see in lysosomes and proteasomes, and energy production and cell death mediated by mitochondria. So in this formulation here, you have metabolites, glucose, glutamine, fatty acids in the mitochondrion, and intermembrane spaces for ATP generation. The tricarboxylic acid cycle intermediates can be extruded to produce lipid and protein building blocks, primarily ATP generation. There's also apoptosis signals for cell death. You have a BAX, BAK complexes, which are used for extruding cytochrome C, which cause a cell death apoptosis, and also in these formulations, cell death necrosis, where you have basically a pH differential, and these are also destroyed, and ischemia and toxin can do this. So the injury effect occurs on a submicroscopic level first, and then you see microscopic evidence, which is usually increased eosinophilia of the cell in question, and this is what we see under the microscope. There's also cellular signaling. These are receptor mediated. These lead to subsequent activities that can act either intracellularly or at a distance. The receptors can be intracellular or associated with the cell surface. Signal transduction, which affects biochemical pathways. This receptor is usually a tyrosine kinase, which mediates insulin, epidermal growth factor, and platelet-derived growth factor. G protein coupled receptors have transmembrane localization. GTP is formed in the inner membrane, and this causes downstream activation of cyclic AMP, which is a messenger. Transcription factors include MYC and JUN. They activate transcription. P53 arrests the growth, and this has implications for neoplasia. Growth factors, these are organ-specific. They have epidermal growth factors, hepatocyte, platelet-derived growth factor, vascular endothelial growth factor, and fibroblast growth factor. The extracellular matrix, interstitial matrix, provides a gel conduit for structure. These are, for example, elastin, hyaluronate, and fibronectin. The basement membrane is a mesh undermining collections of similar cells. You see this in epithelium and in muscle. Collagens, or a triple helix of structural proteins. There are about 30 types of collagen, and they provide components of cartilage, tendon, skin, and scarring. Elastin is used in structures which recover their shape. These are found most commonly in the uterus, blood vessels, particularly the aorta, and ligaments. Proteoglycans and hyaluronans produce a supportive gel matrix, and this allows for tissue compressibility. And then finally, there are adhesion glycoproteins such as fibronectin, laminin, and integrins. These interact with extracellular matrix components. In the cell cycle, we have G1, which is pre-synthetic growth. The S phase is DNA synthesis. G2 phase is pre-mitotic growth. And the M phase is mitotic growth, shown schematically here. So you have the S phase, this is chromosome duplication. This is what you'll see under the microscope. The G2 phase, the M phase is mitosis, and you'll see the chromatin lining up in the middle with the centromeres literally pulling these apart after you get cell division. So you get a daughter cell here and a daughter cell here. We have a G0 rest phase, and then we get a growth in mass, a restriction point, signal sent, and then we get DNA synthesis, and the cycle repeats. This is initiated by cyclins and cyclin-dependent kinases. These are corrective mechanisms. They maintain the correct replicative function. The inhibitors stop this process. These are called CDK inhibitors. Stem cells are pluripotent. They are capable of self-renewal, and they have symmetrical division, where a stem cell gives rise to a differentiating cell and another stem cell. Embryonic stem cells exist in blastocysts. They're tissue stem cells. They're associated with differentiated tissues, and they give rise to cells of that tissue. This is an example of a totipotent stem cell with a zygote, a blastocyst, inner cell mass. Pluripotent stem cells are derived from this in cell culture, and they can be transformed into more specialized hepatocytes, neurons, cardiomyocytes, hematopoietic cells, pancreatic islet cells. You can also have multipotent stem cells, and that lineage can cause endoderm, mesoderm, ectodermal derived cell types. And then you can also find these and used in regenerative medicine. There's a problem integrating the replacement cell into site of damage, such as neural tissue or cardiac muscle. And there's additionally immunogenicity of embryonic stem cells derived from parental antigenic determinants. And the latter can treat the same patient. We call these induced pluripotent stem cells. And that'll be it for the talk. Thank you very much.